Hey there, Bio-30s. Oh, no, wait, you guys are my Bio-20s. Anyways, we are going to keep on going here on digestion. So we have already talked about the mouth, the esophagus, the stomach, and now we're into the small intestine. And one of the main concepts in this unit and a lot of Bio-20 actually is surface area. And so the deal is, how can we increase surface area and get really large molecules broken down into smaller ones so that the enzymes can then work on them and even further break them down so that they're microscopic, so that they can be absorbed into our blood so that our cells can use them. So what is going to increase surface area? Well, in the small intestine, there's little folds called villi in the lining of your small intestine. So here's our small intestine. If we're looking inside of it and we see these little bumps, these are the villi. And believe it or not, each of the villi on each surface of them, if you look closely at the surface, they have thousands and thousands of tinier microvilli. And this is where the enzymes are coming from. And this is where we are absorbing the food into the blood vessels. And so this is a way to increase the amount of surface area for absorption. Okay, so the villi and the microvilli are going to increase surface area so that the enzymes can get at and break down all these different macromolecules. So there's a lot of enzymes we got to know here. And you guys are going to have to be responsible for knowing the main enzymes. Where does it come from? What does it act on? And where is it acting, right? So carbohydrates, carbs, we already learned that they are the very first things in our body that get broken down in the mouth by the enzyme called salivary amylase. Well, coming from the pancreas, we have pancreatic amylase. And so the starch that didn't all get broken down in the mouth, that is now in the small intestine. Well, starch is a very large molecule, a polysaccharide, and this enzyme amylase is the one here that is going to break it down into smaller units called disaccharides, the building blocks. And then the disaccharidases is the next enzyme that breaks them down and breaks down these smaller sugar molecules, simple sugars, into the building blocks, the monosaccharides. So glucose, galactose, fructose, those are the different sugars that are going to be the result of this, the monosaccharides, the single sugars, the simple sugars. And then they get absorbed and they can go into the bloodstream, and they're small enough that now they can be delivered into your body cells. All right, so that is carbs. So large carbohydrates are complex carbs, polysaccharides, starch, are broken down by amylase into smaller units, disaccharides, and then those in turn are broken down into the building blocks. What about proteins? Well, we started protein digestion in the stomach, and we said that proteins were digested in the stomach by pepsin. Now we have a couple of enzymes, trypsin and chymotrypsin, that are coming from the pancreas, and they're going to digest proteins. Remember, proteins are massive molecules. Proteins are hundreds or thousands of amino acids long, and they're all chained and coiled up, primary, secondary, tertiary structure. And so here is where the enzyme chymotrypsin and trypsin are going to break down these really huge molecules into tinier peptides or polypeptides. And then the next enzyme to come along are the peptidases. And these guys are coming from both the pancreas and also the small intestine. And they're breaking down the peptides into the base units, right? The monomers, the smallest particles that we can get broken down into, and that is our amino acids. And then those amino acids can be absorbed, they go into the bloodstream, and now they can be delivered on into the rest of your body. What about fats? Well, we said this is the business end of your guts, guys, right? The small intestine is where all of our different macromolecules are being broken down. And so the first type of digestion that I want to talk to you about is done by this molecule called bile. And bile is not an enzyme. It does not chemically digest fat. What we call this instead is emulsification. And so what is happening is basically the bile is breaking big blobs of fat down into smaller blobs of fat, and we call that emulsification. It's kind of like, I don't know, just you guys have seen blobs of fat when you shake up your salad dressing and it settles out, and all of the fat is at one part of your container. That's the oils, right? And so you know that when you mix it up and you shake up your salad dressing, it's not one big blob, it's smaller blobs. That's all that's happening. This is just physical digestion. It's not chemical. So remember that bile is made in the liver and it is stored in the gallbladder, and then it gets added here from the gallbladder through the bile duct into the small intestine. So now let's talk about chemical digestion. Now we got to talk enzymes. What enzyme are we talking? Lipases. Look at that last part of the name. ACE is typically the ending of an enzyme. So this is how we're breaking fats down, the macromolecule fat, into glycerol and fatty acid. Those are the monomers that break them down. And then this is interesting. They're going into a lymph vessel. So we talked about the lymphatic system. We talked about its role in fluid balance and immunity. Well, another thing that it does is it is going to deliver fat, and the tiny fatty acids are going to be delivered into the bloodstream for circulation to the body. Nucleic acids, that is our last major macromolecule category. We don't really have to worry about those at all. So you can just skip over that part and not worry about what digest nucleic acids. Just know that everything is getting digested in the small intestine, all the macromolecules. For number 7 here, you can go back onto the previous page where I talk about segmentation and peristalsis, and I did that in the last video. It's also in your textbook on page 222. Next page here on hormones, you can actually skip this hormone section. We're not going to talk about this year. But on the next page, there is important stuff on the accessory organs. This stuff we got to know. Okay, so for sure we need to know this. What are the accessory organs? Well, they are organs that are helping with digestion. They're accessories, they're helpers. And the thing is, food does not actually pass through these organs. So they're not digesting anything on their own, but they are helping out. And so the important organs that we have here, the accessory organs, liver is making bile. And again, we just mentioned bile is not an enzyme, just a secretion that is going to physically break down fat by a process called emulsifying. Just tucked in right under the liver here is this little guy, and this little guy is the gallbladder. And that is where we store the bile once it is made in the liver. And then when it's ready, you notice that it has a little tube, a little duct, that the bile can get added into the duodenum. Oops, I spelled bladder wrong. There's an R at the end of that. Okay, so the duodenum, that's the first part of the small intestine. One more super important organ here, and letter C, just into the crook of the stomach here. This little guy, kind of a leaf-shaped organ, is called the pancreas. Now, you might have heard of the pancreas. In fact, you probably have in this class because we've talked about diabetes and we talk about insulin. Well, yes, this is a very important gland for the endocrine system and sugar regulation, but it is also really important in that it makes a couple of secretions, things that get added and help with digestion. So it makes some digestive enzymes. We just talked about some of them. And it also makes sodium bicarbonate, and that is an alkaline or basic substance that helps to neutralize the acid from the stomach. And super important because, again, that stomach acid is very corrosive, right? And when that stomach acid gets into the small intestine, the pancreas has to release the sodium bicarbonate. It's baking soda, essentially, and that neutralizes it, makes it more of a pH level around 7 so that it's not as damaging to the small intestine. Okay, so you guys can read about the pancreas, the liver, the gallbladder, and then we are moving on to the last section of our digestive system here, and that is going to be our large intestine, also known as our colon. And so by the time the food has reached to the large intestine, the colon, there's no more chemical digestion. At this point in time, you're making feces, the waste, the poop, and you're absorbing water, you make vitamins, and now you got to get rid of that waste, that poop, that feces, get rid of it out of the body. So there's no chemical digestion. Is there digestion? Well, it's physical, right? There's peristalsis, you're moving the poop along. And then you move it down into the rectum. That's where you store the feces till you eliminate it. And funny little joke there. It says it has 3 folds to allow you to pass gas and not poop yourself. So, I mean, that's what happens when you fart, right? You can pass gas, you can fart, but you're not going to poop your pants. And then the anus, that is got sphincters, again, those little muscular rings of muscle that act as drawstrings. And so you learn how to control these sphincters when you were being potty trained, right? When you're 2 or 3 years old, your mom and dad, they sat you down. They're like, I'm done with you being in a diaper. And they sat you down on your little potty, and you learn to control these sphincters so that you can go to the bathroom at a certain time and eliminate wastes. Not just any time, not into a diaper, but you have trained that sphincter so that you can release your wastes at a more appropriate time. Okay, so you've got a couple of questions to do here, true and false, matching, some nice diagrams to label, numerical. You guys go ahead and work on that. And as always, if you have questions, michelle.ladine@ghsd75.ca. Talk to you later.